Right. So before I begin today, I was hoping first to start with just a quick poll for those of you who are joining us, really so that we can just better know our audience and hopefully we can better address your questions. So the first, do you have a copy of your child, your loved one's genetic test report with you? Yes or no? And then two, are you here to learn more about genetic test results, really to inform your own results or someone else's results? So I'll give it just a minute or so for responses to roll in. It looks like the majority of you have a report with you, which is great. Thank you for having that out. And I hope that as we walk through various terms that will be helpful. And it also looks like that we're a mix of people who are here for themselves and some who are here to learn more for their child's results. So that's really helpful for us to know. So thank you so much for taking a moment. So to give you a rundown of how this evening is going to go, I'll begin really with an overview of genetics. So for some of you today, some of the concepts and terms might be familiar to you. For others, these might be new terms to you. But really my hope is to review chromosomes, DNA, and genes, to talk about types of genetic changes or variants and the types of genetic testing that detect these variants. We also will walk through some other common terms that you might find on your report and might want to know more information about. And then we'll talk a little bit about the importance of patient data sharing and how that can be helpful in informing our understanding of genetic test results. And we'll end with a Q&A session where I hope we can answer some of your questions. If you have questions as we move through the presentation today, you can feel free to input those into the Q&A box at the bottom of your screen. You also can vote for questions that you would like to see addressed, so you can upvote them, and hopefully we'll be able to make it through questions that we receive today. And also, thank you to those who included questions in their registration survey. That was helpful for us to know going into this evening. So now, starting with an overview of genetics broadly and some of the terms that you might see on your or your child's genetic test report. As I said before, many of you listening have probably gone through the genetic testing process for yourself or a loved one. Many of you might have spoken to a genetic counselor or geneticist, so some of this might be familiar. But my hope is really to break down some of the terms and information today. On the screen here is an example of a report that we'll be walking through. So if you have yours, pull it out. And you can see there are many terms on this report like dominant, heterozygous, de novo, variant of uncertain significance that you might have questions about. And that's exactly what we'll walk through. Each one of us has more than 1 trillion cells that make up our bodies. We have different cells, including our skin cells, brain cells, heart cells, which are really like the building blocks for our body. In each of our cells, all of our genetic material or DNA can be found, and our DNA is really like our body's instruction manual. Each instruction is called a gene, and to keep our DNA, our genes organized in our cells, it's wrapped up tightly into structures called chromosomes. Typically, people have 46 chromosomes that come in 23 pairs. Our chromosomes come in pairs because we get one set from each of our parents. The first 22 pairs of chromosomes are the same in males and females and are organized really just based on size, with one being the largest all the way down to the 22nd pair being the smallest. The 23rd pair of chromosomes are called our sex chromosomes. Individuals assigned male at birth are typically XY. Individuals assigned female at birth are most often XX. As I mentioned, each of these chromosomes is made up of DNA that's wound up very tightly, almost like a ball of yarn. And as I said before, our DNA provides instructions for how our body works and grows, and what those instructions are for are really proteins. Each individual instruction is called a gene, and we each have approximately 20,000 genes that we inherit from our biological parents. Each gene has a specific role within the body. Some determine our eye color, some help our heart develop, others are important for brain development or muscle formation. Every gene, those instructions, are made up of portions that code for proteins, what we call our exons, and parts that are trimmed or cut out before the gene is used to make the protein, what we call our introns. All of our genetic material, all of our DNA is called our genome. And all of the coding portions, those exons that provide instructions, are called our exome. So these might be terms that you see on our report or that you hear your physician or clinician talking about. So just a little bit more information about them. And we'll come back to this later in our chat today too. So if we go back to the report that we're looking at together, we can first see there's a note about gene. So what we just talked about, genes are like our body's instruction manuals. Each individual gene is an instruction. So this particular result tells us about a genetic change in the CHD8 gene. CHD8 provides instructions for a protein that helps regulate the expression of other genes in our body. That's its job and what it does. Our body spells out the instructions in that CHD8 gene and all of our other genes using the letters A, T, G, and C. Our body reads those letters to then make those proteins. When reading the letters within our gene, our body actually reads them in sets of 3. We call this the triplet code, a set of 3 letters in our DNA, in our genes, codes for one amino acid along a protein. Amino acids are like the building blocks of proteins. So our body would read ATG, make one amino acid, CAC, make the next amino acid, and so on until that protein is made. There are 20 different amino acids that make up our protein, all coded by this 3-letter sets. So now we talked about DNA, genes, and proteins in a little bit of a whirlwind, but I wanted to make sure that we all had the same background or sort of knew some of those terms before we move on to genetic variants or changes in our DNA. While most of our DNA sequence is the same from person to person, we all have genetic changes, what we call variants. Some variants can affect how proteins work in the body and cause developmental or health differences. Some genetic changes, variants, are just part of who we are. There are 2 main categories of genetic variants that I want to focus on today, or at least mention today: copy number variants and sequence variants. So copy number variants first are genetic changes where a person has often thousands of extra or missing letters within their DNA. They're large missing or extra pieces of genetic material. If a person is missing a large amount of genetic material, this is called a deletion. If they have an extra piece of genetic material, this is called a duplication. Sometimes I like to think about all of our genetic material, our genomes, being like a shelf of books. A copy number variant means that a person is missing a book or has an extra book, or is missing a chapter, has an extra chapter. Copy number variants are typically named for their location along those chromosomes. You might see on our report that says something about a 19p13.3 deletion, and this really just tells us what piece of genetic material is missing. In an individual that has that on their report, this means that they have a missing piece of material on one of their 19th chromosomes. When we talk about chromosomes, we divide them into parts. We call them arms. So the shorter half or shorter arm is the P arm. The longer half, the longer arm, is the Q arm. So this 19p13.3 deletion tells us on the 19th chromosome, on the short arm, this person's missing a piece, and that 13.3 tells us where in the striped or banding pattern that little piece is missing. Chromosomes have this striped or banding pattern actually when you look at them under the microscope. So this tells us exactly what band or stripe has that deletion in it. The other type of genetic variants that we'll focus in on today and spend much more time on are sequence variants. Sequence variants means that there's a change to the letters within a gene. Often this is a single letter that's different, or sometimes it's a few letters that's different. So if we go back to that analogy of our genetic material, our genome, as a shelf of books, a sequence variant is like there being a spelling change in a sentence in one of those books. Sequence variants are typically named based on the change to the DNA and the change to the protein. If you see a C dot on your report or your child's report, this tells you the change in the DNA. The C is followed by a number, so this tells us exactly what letter is changed in the DNA. So in this example, we're talking about letter 928 in the DMD gene. The first letter here tells us the letter that's typically at that position. At position 928, most people typically have a G. The greater than symbol then is our way of saying is changed to. And the last letter tells us what letter this person has at that position. So altogether, this tells us that at position 928, most people have a G, this individual has an A. This genetic change also can be described by the change in the protein. So if you see a P with a period on your report, this tells you what the change in the protein is. The P gly-310 arginine is our way of describing the change in the protein. The number tells us what amino acid we're talking about. So it tells us that amino acid 310. The first abbreviation is what amino acid is typically there. In this case, it's typically a glycine. Gly is just our abbreviation for glycine. And then the second abbreviation after the number tells us what amino acid is there instead because of this change to the DNA. So in this case, there's an arginine instead of that glycine in this individual. There are several types of sequence variants that I want to mention and talk through. So your C dot and P dot might look slightly different than what we just walked through, and that might be because your type of genetic change is just slightly different. So the first sequence variant, like the one we just walked through, is what's called a missense variant. A missense means that one amino acid, one protein building block is substituted for another based on the DNA change. So that in that example that we just walked through, that glycine at position 310 in the protein is changed to an arginine. One amino acid changed, what we call a missense variant. Another type of sequence variant that you might see is what we call a nonsense variant. Like missense variants, nonsense variants are caused by a change in a single letter in the DNA. What you can see here, typically a G is changed to an A. But in addition to coding for amino acids, the three-letter sets in our DNA also tell the body where to end the protein. So some of those three-letter sets are like stop signs. They say, you're done making the protein, you can stop now. A nonsense variant happens when the DNA change results in an early stop signal, an early stop sign. And this makes the protein shorter than typical, which might not be able to function as it typically does in the body. In the protein change on your report, if it's a nonsense variant, you might see the typical amino acid at that position, so the number, the typical abbreviation here, and then things like TER, stars, and X's are used to denote early stop signals or those nonsense variants. You might also see sequence variants that result in a small number of extra or missing letters within the gene. These are sometimes insertions or duplications that add a letter or several letters to the DNA sequence, like this example here, where the AAC at position 1000 to 1002 is copied. So there's an extra set of those three letters. You also might see deletions on your report, that deletions remove a single letter or several letters from the DNA sequence. Adding or removing letters in the DNA sequence can result in two different types of variants. The first are what we call frame shift variants. As we talked about earlier, the body reads the DNA in sequence in sets of three. If we duplicate a number of letters that's not in the multiple of three, what can happen is our frame of reading can shift. And it can impact the rest of the protein sequence, the rest of those amino acids, and eventually result in a stop signal. You can identify frame shift variants because they typically have FS, short for frame shift, in the description of the protein change. Insertions, duplications, or deletions that aren't in sets of three can cause these frame shift variants. Adding or removing letters in the DNA sequence can also result in what we call in-frame insertions or deletions. So if we delete a set of three letters, like in this example here, we're able to read the rest of that DNA sequence and make the protein. So it results in just a single amino acid deletion. So if a deletion or duplication is in sets of 3, it might just impact a certain number of amino acids, but everything else remains as typical within the protein, what we call an in-frame deletion duplication variant. Some people might also have what's called a splice site variant on their report. So going back to what we talked about a little earlier, as a reminder, our genes are made up of coding portions, what we call exons, and that's what's actually used for those protein instructions, and non-coding portions, what's called introns. Our body removes these introns before using those instructions to make the protein. And there are actually DNA sequences that help our body's machinery recognize where it needs to cut out those introns. There are DNA sequences that might impact these boundaries between exons and introns, what we call a splice site variant. A change to the splicing might cause loss of material that's used to make a protein, so loss of exons, which can cause the protein to be shorter than typical, or it might result in the inclusion of a non-coding portion or typically non-coding portion, which can cause the protein to be much longer than typical. If you see mention of splicing or splice site variant on your report, that's one way to be cued in that this might be a variant that impacts splicing. You might also see it described in ways similar to what's noted here on the slide, which clues you in that it could be a variant that could impact these splice sites. So let's look back at our example report just to walk through another variant. On that report, we see a box with variant and one with coding DNA. So if we look in those two boxes, if we look first at coding DNA, we see a C dot. As a reminder, C dot clues you in to that this is the DNA change. So we have C dot 622, G to A, and that tells us that it letter 622 in the CHD8 gene. Typically, people have a G. This individual has an A. In the variant box, it has P dot, so we know that this is describing the change to the protein. The number tells us which amino acid in the protein that we're talking about. The first abbreviation tells us the typical amino acid at that position, and then the second tells us what's in this individual. So in the protein at position 208, most people have a glycine, but due to this DNA change, there's a serine in this particular person. So a missense variant as well. So now that we've talked about genetic variants, what types of genetic testing might be used to identify these changes? Again, many of you are likely familiar with one or more of these tests from your own genetic testing journey, but I thought it might be helpful to just walk through them because you'll also see some of these terms on your reports as well. The first type of testing I want to mention is karyotype. So we're going to move across this slide from left to right. A karyotype is a test that we used quite frequently in the past, and it looks at the chromosomes for large copy number variants, large missing or extra pieces. With a karyotype, we're actually looking at our chromosomes under a microscope, so we're really looking for big, large changes. If we zoom in a bit more and want to identify smaller copy number variants, this is often done with what's called a chromosomal microarray. So we're still looking for missing or extra pieces. We're just able to pick up things that might be a little bit smaller than what karyotype can identify. If we want to detect sequence variants, we need to zoom in even further to look at the individual letters of our genes. This is done through sequencing. There are a few different testing types that rely on sequencing. So some might look just at a single gene sequencing. Some might look at several genes that are known to cause a specific condition, such as autism or seizures, what we call a gene panel. And then broader testing can include exome and genome sequencing. So exome sequencing looks at all of the exons, those portions that provide instructions for our proteins, while genome sequencing looks for variants in all of your genetic material or genome. Some labs, exome sequencing and genome sequencing, are also picking up missing or extra pieces of genetic material or those copy number variants through that testing as well. So if we go back to the report that we've been walking through today, you might see a section called Test Requested, and that'll tell you what testing was performed. So in this case, exome sequencing is what's listed. So that would look at all of the genes, the coding portions that provide instructions. So after a genetic test is done, a laboratory identifies a variant, what we've just walked through. The next step is to interpret that variant or try to determine if it impacts how the gene works in the body, if it could be associated with a health condition or a risk for a health condition in the future. As I mentioned earlier in our discussion, we all have lots of genetic variants. Some impact our health and development while others don't. When a lab sees a genetic change, they use all of the information available at that time to try to determine if that genetic change might impact health. So for example, this means looking at how the variant impacts the protein. So does it cause the protein to be much shorter? Is it replacing just one amino acid? Looking at where it's located in the protein. Is it a piece of the protein that's really important for that protein's role in the body? Looking at the literature to see if that variant has been seen in other individuals with symptoms before. Looking at population databases to see, has this particular genetic change been seen in healthy people without symptoms? And then they put all this information together to determine their current classification, or based on the current information, if they feel like this variant is associated to health differences. Classifications fall into three main buckets. They are benign, uncertain, and pathogenic. Benign means that the copy number or sequence variant is not known to cause any significant health or developmental differences. Pathogenic means that it's a copy number or sequence variant that's known to cause symptoms or features, or put people at risk for those conditions or features, or maybe they're a carrier for the condition. You might see terms like likely pathogenic or likely benign. Those fit into those main buckets. It's just we use a few extra categories when classifying these. Finally, if there's not information to put a variant into the pathogenic or benign category, it's often called a variant of uncertain significance, or VUS. This means that based on the information we have available, there's really not enough evidence to say that a variant is associated with health or developmental differences or is not associated. More information really is needed to help better put it into one of those clearer categories of benign or pathogenic. Sometimes additional information might be things like testing other family members. Other times we need to wait just for more information to become available to help really inform our understanding. So if we look back at our report, we see that this variant in CHD8 was interpreted by the laboratory as a variant of uncertain significance. So again, this means that it's uncertain at this time. We don't have enough information to say if this particular variant is impacting how the gene works in the body and is associated with this patient's developmental history. Again, this snapshot of classification is just based on the available information at the time. So if you have a VUS, we often encourage you to reach out to your clinician or healthcare provider to check in about the results periodically, maybe every 1 to 2 years, because as new information becomes available, we do hope to move them into more definitive categories over time. So now on to some other common terms that you might see on your reports. So on your reports, you might see terms like autosomal dominant, autosomal recessive, and X-linked. To walk through these, I wanted to return to this slide that we looked at earlier today. As a reminder, we all have 2 copies of the first 22 pairs of our chromosomes. Again, we have 2 copies of each because we get 1 from each of our biological parents. So for any genes found on these 22 pairs of chromosomes, we have 2 copies, 1 from each biological parent. Our last set of chromosomes, again, are our sex chromosomes. Girls, typically XX, boys, X and Y. So girls, for any genes on the X, have 2 copies, while boys just have 1 copy of the gene, most of the genes on the X chromosome. Autosomal dominant and recessive are used to refer to conditions caused by changes to genes that are located on those first 22 pairs of chromosomes. Again, we typically have 2 copies of each of these genes. Conditions that are caused by just having a single variant on one of the copies, so having a single variant is enough to cause symptoms or increase the risk, is what's called an autosomal dominant condition. One change is enough to cause that condition. If pathogenic variants on both copies of the gene are necessary to cause a condition, this is what's called autosomal recessive. Conditions caused by pathogenic variants in a gene on the X chromosome are what we call X-linked. In X-linked conditions, symptoms can differ between individuals who are assigned male at birth and those assigned female, since females typically have 2 X's while males typically only have 1 X. Sometimes autosomal recessive, autosomal dominant, and X-linked are called inheritance patterns, but they don't tell us if a genetic variant was inherited from a parent or whether the variant was found on one or both copies of the gene. They're really there to help us understand if 1 or 2 pathogenic variants are needed to cause a condition and whether the gene is on the X chromosome. So if we go back to that report that we looked at, CHD8 is associated with autism spectrum disorders, in those with pathogenic variants. And in those with pathogenic variants, it's inherited in an autosomal dominant fashion. So having just one change on one copy of the CHD8 gene is enough to cause those symptoms. But again, in this particular patient, it doesn't tell us if their variant was inherited or much more than that. It just tells us really about this particular gene and how, when it does cause symptoms, it's passed through the family. The words heterozygous, homozygous, and hemizygous, which you might see on your or your child's report, tell us about the specific patient that was tested. If the variant was found on, it tells us if the variant was found on one copy or both copies of the gene. So heterozygous is used to describe a person who has the variant on one copy of their gene. Homozygous is used when the patient has the variant on both copies of their gene. And hemizygous is used to describe males who have the variant on their only X chromosome. So they only have one copy and they have the variant on that particular gene. So if we go back to the report that we've been walking through, zygosity is sort of the bucket for these terms, and this individual is heterozygous. So that tells us that they have the CHD8 variant on one copy of their gene. Testing parents can help us know if a genetic change was passed from a parent onto their child. If a variant was passed on from a parent to a child, it's often said to have been inherited. And you might see words like maternal or paternal on your report. Maternal means inherited from biological mom. Paternal means inherited from biological dad. Some genetic changes also can be new and unique to a child or an individual, meaning that they weren't inherited from a parent, but happen early on in sperm or egg or during development. Whenever a genetic change is thought to be new and unique to an individual, we call these de novo. De novo actually means anew, so it's new and unique to them. So if we go back to our report, the inherited from box on the report that we've been looking at tells us that this particular variant was found to be new and unique to this individual. You see the de novo listed there. So that means that they tested parents. It wasn't found in either of their samples. So we feel like it's new and unique to this child. Finally, a few families or individuals might have the words mosaicism or mosaic noted on their report. Mosaicism means that only some of a person's cells have the genetic variant. This means some cells have the variant, others do not. For some families, this can be identified through their testing, but our testing technology doesn't pick up all cases of mosaicism. But when it does pick it up, it means that we know that some of the cells that we tested have the change. Sometimes a parent might have a specific type of mosaicism called germline mosaicism, which means that some of a mother's eggs or some of a father's sperm cells, their germline, have the genetic variant. Our current technology doesn't allow us to detect germline mosaicism, but it's usually identified when parents have more than one child with the same genetic variant, but them themselves in their blood or saliva samples that were tested don't have the variant. If a variant is thought to be de novo in a child, we often say that the chance for germline mosaicism is about 1% in that family, though it does vary for some specific conditions. The chance that family members will have the same genetic variant really depends on the inheritance pattern within the family, whether the variant was inherited or de novo. So putting all those pieces together, your genetic counselor can often walk you through what are the chances that someone else will have this variant, this specific condition as well. So now that we've walked through some concepts and terms on your reports, I wanted to briefly mention the importance of data sharing and how it can really be used to inform our understanding of genes and variants before we move on to the Q&A. So as some of you may know, myself, Erin, and Alexis on the call all work as part of GenomeConnect, which is a broader piece of the clinical genome resource. ClinGen, or the Clinical Genome Resource, is an NIH-funded project that's really working to better understand genes and variants and their relationship to health. And the end goal is to ultimately improve patient care by better understanding genes and variants. ClinGen recognizes that data sharing is really important to increasing our understanding, and for that reason, started GenomeConnect, a patient registry to help patients share data. ClinGen encourages data sharing through GenomeConnect, as well as encouraging data sharing from other stakeholders, including clinicians, labs, and patients as well. The purpose of GenomeConnect is to share data with databases, including ClinVar, which is a publicly available database that holds de-identified information about variants and its relationship to human health. ClinVar is public and freely available, and it's really a major resource for healthcare providers, labs, and researchers who are caring for patients, those who are performing testing and researching genetic conditions. The database itself is submission-driven, meaning that it relies on labs, clinicians, and others to share data. And so this is where GenomeConnect shares information from participants who are interested in data sharing, because ClinVar is really how we pool resources and knowledge in an effort to advance our understanding of genes and variants. Ultimately, data sharing is important because it can help increase our understanding of genetic conditions and can help doctors and genetic testing laboratories better understand genetic changes that cause disease. This increased understanding can help identify interventions and treatment, can help other individuals get clear genetic test results in the future. So data sharing really helps us get to that end of better being able to care for patients. So from our experience within GenomeConnect, that patient registry that allows individual patients to share their genetic and health information, we've really seen that patients can contribute valuable information to move our understanding forward. In the first way, patients can often contribute additional health information and other details about their history that can be incredibly valuable in answering our questions about genes and variants. Labs and clinicians might have health details, information about whether a variant was inherited in a family, but they might not be able to include those in their ClinVar submission just because of the nature of how they submit. But patients who are engaging in data sharing through GenomeConnect are providing this information in the public space. Additionally, we've seen from our data sharing to date that patients choosing to enroll in GenomeConnect are also providing genetic changes that haven't been submitted to the ClinVar database before. From our submissions to date, 41% of genetic changes weren't submitted to ClinVar from the laboratory that did the patient's testing or anyone else who's submitted to ClinVar previously. So putting that information out there in the public space can be useful the next time a genetic variant is identified or if similar variants are found in other patients. To help illustrate how patient data sharing can provide valuable data to increase our understanding, I just wanted to provide a real life case from our experience with GenomeConnect so far. For the purposes of our discussion today, I will call this patient Ella. Ella was a pediatric patient who had a history of seizures beginning when she was just a baby. She had exome sequencing, so that genetic testing that looked at all the coding portions of her genes back in 2015, and had a likely pathogenic variant in a gene associated with seizures found. So this gene was found in her testing and thought really to be her answer for her seizures. Her parents were tested. The variant was not found in either of their samples, suggesting that the variant was de novo, or again, new and unique to Ella. Ella's family had an answer for her seizures, an explanation for her history, but they chose to participate in data sharing, I think recognizing that this information could be helpful or useful to other families. We submitted Ella's de-identified genetic information to ClinVar. So for GenomeConnect participants who provide their health history via surveys and upload a copy of their genetic test report, our team pulls out all of the important information and de-identifies it. So no name, birthday, or other identifiers are shared. So we submitted this on the family's behalf to ClinVar, and we saw that there were 2 other records in the database. The lab who had done Ella's testing had also submitted the variant to the database, but they had really fewer details about her clinical history. So they didn't have anything about her seizures, also didn't have the information about her parents' testing, that it was new and unique to Ella. A second lab, Lab 2, had also submitted the variant to ClinVar, but based on the information that they had in 2014, they felt like the variant was uncertain. Based on what they knew, they didn't feel like they could put it into that pathogenic or benign bucket. So after our submission, Lab 2 reached out to our team to ask more about this participant, and specifically were interested in the de novo status, or that we had, in her family, her clinician had been able to test mom and dad. In Lab 2's family, they weren't able to test parents, and sometimes testing other relatives can be really helpful in better understanding uncertain results. So we were able to tell them a little more about our participant, that her variant was new and unique to her, which allowed Lab 2 to go back and update their classification for their patient. So they were able to say, based on that another individual has this genetic change, based on how it was inherited in her, all of these pieces, that's enough for us to go from uncertain to likely pathogenic. So Ella's results in care weren't impacted by her family taking the time to share her results, but ultimately them doing so enabled another family to get clear genetic testing. So in addition to contributing information to inform our understanding moving forward through the registry, another benefit back to participants is that they can choose to receive updates about their genetic test results if we should come across them from our data sharing. As I mentioned before, variants are classified when they're reported just based on the knowledge base, but over time our understanding might shift. So for example, that VUS from Lab 2 in the last example, a couple of years later, was changed to likely pathogenic. Through our data sharing, GenomeConnect might become aware of shifts in classifications. So as part of participation in the registry, we give participants the option to receive that information from us as part of the registry. So if in cases where we do identify a change to classification, we would circle back with participants that choose to receive that through the registry. So finally, before we move to Q&A, I wanted to also just show briefly a sample ClinVar record, because I think one of the questions that we get a lot is, well, what does my information look like in ClinVar if you're sharing it with the database? So ClinVar records look a little bit like this. Any variants that have been submitted, it has some of the things that we've talked about on today's call. So gene name, C dot, P dot, and then if you were to scroll down in a record, you'd see that for this particular submission, this is from GenomeConnect. So this is something that we submitted on behalf of our participant. And there's information in here about how the variant was classified by the reporting lab, when it was classified. That inheritance information is all listed here, but no identifiers or identifying information. In addition to the variant information, we also include information about participants' health history, included just as structured terms. So again, nothing identifiable in that way. So if you're already a participant in GenomeConnect, thank you for participating in data sharing. If you're interested in participating in data sharing and have yet to enroll, I did put the link here on the slide, genomeconnect.org. And we welcome you to to sign up online if that's something that you're interested in doing. We do think that data sharing is incredibly valuable, and our experience to date has really been that patients are an important piece of that puzzle, that they're contributing useful information to help us better understand genetic changes over time. So with that, I think we have about 15 or so minutes to answer questions from the audience. So if you have a question, feel free to use that Q&A box. And I think we can see what we might have.
All right, thanks, Jules. We've had a few questions come into the chat about specific variants, and I don't want to read anyone's specific variants on this recording just to not give away any of your sensitive information. But I'll try to ask this one in a way that's not doxing in any way. But if you have a variant in a gene and it was called intronic, heterozygous, not observed in a database and marked as benign, how would you describe that? What might that indicate?
You said not.
I'll repeat, intronic, not observed in database, marked as benign.
Okay, let's break those terms down a little bit. So let's start with intronic. So you'll remember, again, our genes are made up of exons and introns. So exons are pieces that code for proteins, introns are non-coding. But one piece, so intronic first tells us that this particular variant is in one of those introns. Sometimes intronic sequences can still be important because of that splicing piece of the puzzle that we talked a little bit about. So those, when our body sort of removes introns, it looks for certain sequences in introns to know what introns to remove or where introns are. So intronic tells us that this particular sequence is within an intron. A clue that you might see on your report, if it doesn't say intronic, if there are pluses or minuses in the C dot, that's often a clue that it's an intronic change, so something within an intron. So then you said not present in a database. So that—
Yes, what does that mean?
Great question. So there are a couple of different databases that the report might be referring to. So one is ClinVar. So the database that we submit to through GenomeConnect, that laboratories submit to, that we really use as a repository of variants, that might be one database that they're referring to, that it hasn't been submitted there. Another database or type of database that we think about are what we call population databases. So we have some resources that have looked at the general population for genetic changes. So if it's not present in a population database, a report might note that as well. That piece can be helpful because if we've seen a variant in many people in the general population, that might be a clue that it might not be associated with a health difference or disease. Or if it's absent from the population, that might be a little bit of evidence towards, well, maybe this could be doing something. So that's another piece of the puzzle that the laboratory might be putting together to classify it. And then the word benign, I think, came up, Erin, so hopefully I'm hitting on all of these, is that classification. So based on all of the available information, what does the lab think of that particular variant's role in health and disease? So benign, as a reminder, tells us that based on the information we have, that particular variant has not been associated with a genetic disease or risk for a genetic disease.
All right. We've got another question coming in, someone who is a participant in GenomeConnect and said that they have submitted 3 of their genetic tests to us. So if a new result comes in through ClinVar, will I be notified? And is that automatic? Does the computer spit it out? Or can you tell us a little bit more about that process?
Yeah, that's a great question. So thank you, first of all, for sharing your results with us. We appreciate having you in the registry. So the first piece is that this is something that you choose to receive updates from GenomeConnect. So within the contact and sharing preferences within your account, there's a question about, do you want to receive updates from us? It's a question that you likely completed upon enrollment, if you've enrolled in the past couple of years. But I always encourage folks to check those preferences databases and make sure that they're up to date. So if you've selected yes, that you'd like to receive an update from us, if we come across an update through ClinVar, we would share that back to you via email. So how do we identify updates through ClinVar? So after we submit, we have a monthly check of all of our submissions against the other submitters within ClinVar. So if we see that, just to use a laboratory as an example, if GeneDx has reported that variant to you as uncertain back in 2018, and next month we look and we see that GeneDx has now submitted that to ClinVar as benign, that's something that we would feed back to you. Now, because we're not a medical provider, we want to make sure that we give you information and then you circle back with your healthcare provider to learn more. So our email would tell you a little bit about what we found from ClinVar, but would encourage you to reach out to your clinician to confirm that this truly is an update, to get an updated report, and to make sure that any impact on your care, discussion about your results, your provider who's cared for you can have those discussions.
Thanks, Jules. We got another question, and I'm actually looking for a resource to send this person in the background. What do the letters R and X mean in the variant P dot something, something, something? You talked about 3-letter abbreviations. Can you talk about 1-letter?
Yes, I need to update my slides, include 1-letter abbreviations. So thank you for this question. So amino acids, we have 20 of them, and they can have 3-letter abbreviations or 1-letter abbreviations. So as if genetics wasn't complicated enough, we have to go and complicate it further by having different abbreviations for the same thing. So the amino acid arginine can be abbreviated with ARG, the 3-letter abbreviation, or just the letter R. So in that example, that was telling us that the typical amino acid at that location is an arginine. And then the X is one of our ways of noting a nonsense variant. So we can see in the protein change X's, stars, or TER, short for termination. All of those are our way of noting that this is an early stop signal. So remember that those 3-letter codes in our DNA can code for amino acids or can code for a stop sign to tell your body when to stop making a protein. So that would tell us that that DNA change took it from an arginine to a stop sign. Great question.
Yep, and I put, hopefully everyone can see the Q&A, but I just put a link. This is a very common thing. You can just Google 3-letter to 1-letter amino acid abbreviations. I just put the first one I saw. These are very standard, common things. So there's a handy table in the Q&A if you are interested in that. Another question for you, Jules. So can you tell us, are VUS the only classifications that can change?
Great question. So no. So VUS, we ultimately hope to move into a more definitive category. So those are the ones that we hope change over time. But even other classifications we know can shift. It's less likely for something that's in a definitive bucket of pathogenic or benign to shift, but we know that it does happen. Genetics, we're constantly learning, and new information, new resources come available, and sometimes those categories can shift as well.
If you wouldn't mind, let's go back to our little example report. There are 2 other fields that I would love for us to just spend a moment on, just for people to notice. So, well, one that you have blanked out here, but let's talk about the date of the report and how that might impact what you think about the variant classification, particularly as new classification procedures have come out over time.
Sure. So yep, this is not a real humans report, so I could have put things in here, but just kept it blank. So your date of report is going to be somewhere on the paper that you have. Often it's in the header, somewhere up here. So in this example, here. Other times it might be on the last page under a signature of whoever signed out your genetic test. So 2 places to look for it. And this will tell us the date that this variant was classified. So as we've talked about this evening, these classifications are a snapshot in time based on whenever that report came out, the available evidence, how it was classified. And we know that some things have shifted over time. So population databases, which we talked a little bit about, those were very small just a few years ago and have gotten much larger. So much more detailed data to compare to. How we think about genetic changes and how we put them in these classification buckets has also shifted over time. And we've gotten better and better guidelines for how to classify variants and what pieces of information to put together and what might be more clearly push us towards pathogenic or benign. So your date of report is important because it gives you a sense of when was this variant classified. And as I said before, over time, things can shift, and VUSs we certainly hope shift. So we do encourage folks to circle back with their providers periodically just to make sure that, okay, this was in 2012 or 2015. Let me check in on the current knowledge base. So VUSs, we often say every 1 to 2 years, but I think it's good practice to check in with your provider of, is there anything new from their perspective, and has anything shifted since the date of report? Anything else you'd add there, Erin?
Yes, I mean, that was the sentiment that I wanted to get across, that if your report is older, like Jules said, population databases were not even available, say if you had your testing done 15 years ago, 20 years ago, et cetera. So definitely things are changing all the time. So if it's been several years since you've had your testing, particularly if your result was a VUS, it is worth it to check back in with your healthcare provider to see if anything's changed. The other thing I wanted to just touch on, can you talk about the disease that's in the box? Not the indication, but the disease that's in the box. What does that mean? That the person has a clinical diagnosis of whatever that disease is, and how do the labs decide what they put there?
Great question, Erin. So disease is often the disease that's been associated with that particular gene. So it doesn't tell us, it certainly doesn't tell us if this individual has this particular condition, but it's just that particular gene based on the information we have, what condition has it been associated with. So CHD8 in this example, for those with pathogenic variants, has been associated with autism spectrum disorder. It doesn't mean that everybody with a pathogenic variant has autism spectrum disorder, but it's the gene disease association that we've seen before. Other genes might be linked to a muscular dystrophy or to an increased risk for cancer, but it's just based on that gene. You need to look a little bit further to understand this particular variant and its classification. Gene disease relationships are really based on what's out there in the literature. So what's out there in publications, what's been described before. And ClinGen has efforts to try and define these gene disease relationships and better understand what are the conditions that genes are tied to. Now, there are some genes that are out there that haven't been associated with the disease just yet. So on your report, you might see none currently described or none in that disease box. And what that means is that that gene hasn't been associated with a health condition or anything just yet, but maybe based on some preliminary evidence or just some things that we know about the gene, it's something that we want to keep tabs on, or the laboratory wants to report out. It might be, if we're thinking about someone has testing for a muscular dystrophy and has a gene that is expressed in muscles and seems to be important for how those muscle fibers come together, but hasn't been associated with a clear condition yet, the lab might report that on exome sequencing and say, none currently described yet, but it's expressed in the muscle. So it's uncertain, we need more information, but they report it out to folks.
And I would just add that there are entities out there that kind of come up with these standard disease names, and a lot of times laboratories will use those. And it made me think about that, Jules, because we have gotten emails from participants before asking, like, why does my report say this condition? I don't like that name. That doesn't describe my child. So I think it's just important to know that laboratories are often picking from a predetermined set of condition names. It's not that they're saying that your child has XYZ or what have you.
Exactly. That's a great point, Erin. And it's just something to keep in mind.
And then we have one final question in the chat. Why do we sometimes see that a variant is related to more than one gene, or if we see the abbreviation LOC, what does that mean?
So variants, in particular, copy number variants can include multiple genes. So if there's a large missing or extra piece of genetic material, it sometimes includes more than one gene. So that might be one reason that on your report that you're seeing multiple genes listed, or that it's not just one, but a few. And then LOC, Erin, would you? So I know LOH. I want to make sure I have.
Yeah, so LOC is often used to describe like a locus where we think there's a gene there, but we don't know enough about the gene to give it an actual what we call HGNC gene symbol, like that CHD8 that you see on the screen. That's an official gene name. But sometimes when, genes we're not really exactly sure what they do. We're not even sure if they are real. We just refer to them by more generic names like LOC, number. And that just means we really don't know much about what this thing is yet.
And then that just helps us know where in the genome is that piece. So locus ID kind of helps us know where along those chromosomes in the genome is it located. So locus, location, where is it?
Right. And one quick last question. If your variant is named like P amino acid number and then equals at the end, what does that mean?
These are great questions, and my slide deck's just going to get larger now for including all these things. So the equal sign just tells us that that particular DNA change did not change the amino acid sequence. So you might have if we look at our example here, a G to A. So the DNA has shifted, but ultimately, if there's an equal here, that would tell us that it's still a glycine at that amino acid. So that amino acid sequence didn't change. DNA is different. Protein remains the same. Sometimes we call those silent changes. So you might see that on your report as well.
Or synonymous changes.
Wonderful.
All right. Well, there are no remaining questions in the chat. We are right at about the end of the time, Jules.
Perfect. So I just want to thank you all for attending today. And this was our first webinar, so we haven't done this before. So we really love your feedback. So I'm going to just pop a link in the chat. So a Google form where we can just collect some information about how did you think this went? What could we do differently? If we did this again, what additional topics might be useful? So if you would like to provide feedback, we would welcome it because we hope that it could shape additional events like this in the future. I want to thank Erin for moderating and Alexis for being here as well. And to thank all of you for taking some time out of your evening to join us and to really ask some really wonderful questions. So thank you so much.